Have you ever looked at a massive field of daisies and wondered how on earth scientists figure out how many there are? I mean, they don't count every single one, do they? Well, you're in the right place, because in this explainer, we are going to master quadrant sampling.
Okay, first up, let's really get to grips with the huge problem that ecologists have to solve whenever they step into a habitat.
So just imagine your teacher says, right, go and count every single daisy on the school field. You'd just look at them, wouldn't you? Where would you even start? Counting them all one by one isn't just a bit tricky, it's pretty much impossible. It would take you absolutely ages.
And that's the thing, it's just not a practical way of doing things. This is exactly why scientists came up with a really smart technique called sampling. The whole idea is, instead of counting everything, we just count a few small sections and then use that data to make a really good guess, an estimate, for the entire area.
Now, before we get sampling, we need to get some key terms sorted. Let's build up the picture. Think about the whole field. You've got the grass, the daisies, the soil, the sunlight, the rain, all of those things, the living or biotic parts, and the non-living or abiotic parts, interacting with each other. That's your ecosystem.
Okay, now let's zoom in a bit and just focus on the living things in that ecosystem. We call that the community. It's all the different populations of all the different species living together. So that's your daisies, the grass, the clover, the worms, maybe some bees buzzing about. You get the idea.
And if we zoom in one last time and just look at one of those species, in this case our daisies, we're now talking about the population. And it's the size of this population that we're trying to figure out.
So we know why we're sampling and we know what we want to measure. The big question is, how do we actually do it? Let's break the method down step by step.
The method for random sampling is really logical, and it's all designed to make sure our results are fair. First, you'll lay out two tape measures at a right angle to make a grid. Then you'll use a random number generator on your phone or calculator to get a pair of coordinates. You walk to that spot, pop your quadrat down, and carefully count the number of daisies inside the square. Write it down, and then you repeat. The more times you do it, the better your data will be.
Now let's just pause on that word random. Why is that so important? It's all about avoiding bias. You see, it's just human nature to look at a field and think, ooh, there's a big patch of daisies over there. I'll put my quadrat there. But if you did that, your results would be completely skewed and your estimate would be way too high. A random number generator takes you out of the equation. It makes sure every single part of that field has an equal chance of being picked. That's what makes the result scientifically valid.
You've got a page full of numbers. But what do they actually mean? This is where we turn those raw counts into a proper population estimate.
So let's imagine this is our data. We've thrown our quadrat 10 times randomly, and these are the number of daisies we counted in each one. As you can see, the numbers jump around a bit, from 4 all the way up to 9. That's totally normal, and it's exactly what you'd expect to find in nature.
We need to find the average, or as we call it in science, the mean. It's really simple. All you do is add up all the daisies you counted, and then divide that total by the number of times you threw your quadrat.
So for our data, if you add up all those numbers, you get a total of 60 daisies. We took 10 samples, so we do 60 divided by 10, which gives us a nice simple mean of 6 daisies per quadrat.
And here's the final step. This is the really clever bit where we scale everything up. We know that, on average, there are 6 daisies in each of our quadrats. So how do we work out how many are in the whole field? Well, we just need to know the total area of the field. Let's say it's 20 meters by 50 meters, which is 1,000 square meters. We just multiply that total area by our mean. So 1,000 square meters multiplied by our average of 6 daisies gives us a final estimated population of 6,000 daisies.
Random sampling is fantastic for getting an average number across a whole, fairly uniform area. But what if you're not interested in the average? What if you want to know how the number of daisies changes as you walk from the shady bit under a tree out into the full sun?
For that kind of investigation, you wouldn't use random sampling. You'd use something called a transect. This is where you lay a tape measure in a straight line across the area you're investigating. Then, instead of picking random spots, you place your quadrat down at regular intervals along that line. This is called systematic sampling, and it's the perfect tool when you want to see how a species is distributed along an environmental gradient, like light levels or soil moisture.
So let's have a think about how we can evaluate this practical.
To really get those top marks in an exam, you have to think about this stuff. How could you make your results more reliable? Well, the easy answer is always to take more samples. The bigger your sample size, the more confident you can be in your final estimate. Did you avoid bias? Yep, you used a random number generator, so tick that box. But what about the limitations? Well, what if the daisies grow in clumps? It can be tricky to count them. And this method is totally useless for animals that move around, right?
What are the 3 absolute must-haves to get the marks?
Number 1, nail the method. Describe it clearly, step by step, and always mention using a grid and random numbers to avoid bias. That's a key point. Number 2, show you're working. If they give you data, don't just write down the answer. Show them how you calculated the mean and then how you calculated the final population estimate. And number 3, evaluate. Be ready to suggest how you could improve the investigation, usually by taking more samples, and be prepared to discuss the limitations of the data. Get those 3 things down and you'll be flying.
And that's it. That is the power of quadrat sampling. You've now got a really fundamental tool that ecologists use all the time to investigate the world around us. And it allows us to start asking even bigger, more interesting questions. You know, why are there more daisies in one part of the field than another? What factors, like the amount of sunlight or water or competition from other plants, are controlling where they live? Keep asking those why questions, because that's when you really start thinking like an ecologist.